We are also seeing some new stuff from another different research line that we have here in our lab. And feel free to stop me to ask anything you want. There are some slides that I will skip because we had already seen yesterday, or because they are not too important for the scope of this class today. So this is, again, talking to you about the nanoscale, or I mean the length scale, and the nano region of this nanoscale, and what kind of system you are looking at or looking for. So proteins should be something around here, but this slide was prepared to nanomaterials. So here we are worried about, for example, let's say inverted micelles or silver or gold nanoparticles, liposomes that we are talking a little bit, cubosomes. There is another lipidic-based nanoparticle that we will talk a little bit, and also some big stuff which are not the topic of this talk today. These, again, depending on the kind of system you have, you must choose the proper experimental technique. I already saw that to you yesterday. Don't worry, I will get all of those slides in PDF to the guy here, so you have access to everything, so you don't need to get any note regarding any particular formula or something like this. You can feel free to just pay attention in the slide, because you have access to that perhaps this afternoon or tomorrow. So, when we are talking about lipidic-based nanoparticles, we must remember a very, very special kind of molecules that we have in chemistry, which are called amphiphilic molecules. Those are kind of special because they are composed mainly by, in a very, very general way, two different regions. One of the regions of the molecule is hydrophobic, which means it does not like to make, actually it does not, the molecule doesn't like, water is not able to make hydrogen bonds with that particular region of the molecule. So water does not like to be in touch with that molecule, because water wants to make these hydrogen bonds every time. And so water does not like that region. And you have the hydrophilic region, which are that region that water is able to make these hydrogen bonds, or even it could be, for example, a deuterion, or a cationic or anionic polar molecule. Doesn't matter for us, but what matters for us is that water likes to be in touch with that region, and water does not like to be in touch with the other region. Those are called the amphiphilic molecules. They have a very, very broad spectrum of a molecule, and there are several researchers working on this from decades, perhaps even a century or so, and generally we represent a molecule with something like this. We have this, what we call the polar head group, which is the polar group, which means it's the group that likes that water likes to be in touch with. And we have the hydrophobic group, which generally are composed by carbon and hydrogen in such a way that it's not able to make hydrogen bonds with the water. So water does not like to be in touch with that guy. Now we have when we are talking about the self-aggregation process, this is an entire energy-driven and entropically driven process. We have some theories regarding this. Let's talk about the micellization theory, because I think it's the best one to understand what is going on here when we have this self-assemble. So if you are putting these molecules in water, inside the water, up to a certain concentration, it's fine. Water does not like to be in touch with that, but you don't have so many molecules, so we can survive like this. However, if you start to increase the molecule concentration, you will reach a point, what we call CMC, if you are talking about micelles, or CAC, critical aggregation concentration, or critical micellar concentration, depending on your case, that it will be not energetically favorable to the water being in touch with those several hydrophobic regions. So water somehow try to organize all of those systems spontaneously, and organize them in such a way that it increases the water contact with the polar regions and decreases as much as it can the water contact with the hydrophobic regions. This is called the micellization process, or the self-assemble process. And it's very funny, because the structure that you may have using those molecules, those amphiphilic molecules, can vary a lot. And there is what we call this, let's say, empirical parameter. We call the packing parameter, which is the volume of the hydrophobic region divided by the length of the hydrophobic region times the cross-section of the hydrophobic region. And depending on the value of this guy, you may form spherical micelles, you may form cylindrical micelles, you may form infinite planes by layers, you may form inverse hexagonal structures, you may form cubic structures. There are several different studies that, or several different forms, several different shapes that you may form using this type of molecule, depending, of course, on the molecule that you are using, depending in some content also on the physical-chemical properties of your system. For example, if you are studying an anionic surfactant in pure water or in water in the presence of salt, you can change this kind of behavior, this kind of structure. Instead of something like this, you can go to something like that. All of those stuff is possible. In this other slide here, we have a more genetic, a more complete description of all of the possible structures. If you are interested in that research area, there are several reports in the literature. There are several books in the literature describing the physical-chemical processes involved in the self-assembly, the physical or the chemical process involved. So I will not stay too long talking about this. You may find several reference books around it. You can ask me in the end of the class. We can talk a little bit about them. Today, I choose to talk to you with two different molecules, which is monoline and phytantriol, and both of them they have a very particular property because they form cubic structures in solution. In excess of water, spontaneously, it forms a cubic structure, mainly one of these three cubic structures depicted here in this slide. I will later on talk a little bit more about this, but there is a very delicate interbalance between the hydrophobic effects and the hydrophilic effects to perform this kind of minimum surface in which our lipid self-assemble. I'm talking about them because this is known from if I'm not wrong, from '68 in an article of Professor Vittorio Luzzati. He was in that period in Grenoble, and he talks for the first time the possibility of amphiphilic molecules to self-assemble into cubic structures. And he has a very beautiful paper describing the electron map dance. It's a very beautiful work. So we are talking about the early 60, 65 or something around this. And he uses the derivatives of monoline. But those are infinite structures. Later on, around 20 years ago, someone discovered that if you mixed this guy with polymers, you were able to make a nanoparticle with a very high hydrophobic volume. The main advantage to use this kind of molecules is the hydrophobic content that you have access, and we are talking more about them later in the lecture. Only you to understand that these are the kind of structures that you may have. We have water tunnels, and between two adjacent water tunnel, let's say this one blue and this one red here, we have a kind of a bilayer in a minimum surface arrangement. Don't worry, it's very difficult to understand how are these organized. I forgot to bring it today, but we even constructed some kind of sketch to try to understand better how are they organized. It's a very delicate balance between these structures, so you don't need to worry about them. What you are seeing here is the surface, is the end of the tail of each lipid. For example, if this one guy here is from phytantriol, you have to one side of the lipid, for the upper side, one molecule, and for the inner side, the other molecule. And this forms the bilayer that I'm talking about. But later on, I think I have a better slide. Just to remember, yesterday we talked a little bit about small angle scattering. We use small angle scattering because the sizes of this cubic structure are more or less compatible with Sachs technique. We are talking about latency parameters of 15 nanometers, 10 nanometers, so it's very nice to use X-ray to try to better understand and to characterize this kind of system. So you'll not take some time, and we already talked about this yesterday. And this is the point that I would like to talk a little bit a little bit to you right now, because every time you have a repetition in space, in some particular system, could be one-dimensional. In the case of a multilamellar stacking, you have a one-dimensional crystal. And we like it because in physics, we like to simplify everything. When you are starting to study solid-state physics, you start to make the calculations for the crystals. And you say, okay, let's suppose a one-dimensional crystal. You say, come on, crystal, one-dimensional, we don't have in nature. But then we discover that we have one case, at least this one. And it's fine because we can do all of the math, and it's analytical, to one dimension. Then we have a two-dimensional lattice parameters, let's say a bench of cylinders aligned with each other. They have this kind of symmetry. And the three-dimensional, that we don't know all the math yet, which are the case of the cubic structure. Every time you have a crystal, or you have something that's repeating in the space in 1, 2, or 3 dimensions, you may have, depending on the distance in your experimental setup, peaks on your scattering curve. Those peaks have very, very, very specific positions to be. It cannot be in any position as they want. They must respect what we call the Brag's law. The Brag's law is the main law from X-ray diffraction. It was developed in 1920, more or less, by the Brags, the Bragg father and the Bragg son. And they won the Nobel Prize due to the Bragg diffraction theory, if I'm not wrong, 1925 or 1924, something around this. And the Bragg son was the youngest researcher, to receive the Nobel Prize up to 3 or 4 years ago, when we have the Peace Nobel Prize won to Malala, that Pakistan girl that was suffer a lot of tragedy because she would like to study and so on. And she was laureate with the Peace Nobel Prize. Then she became the youngest one. Up to that day was the Bragg son, the youngest one, with the diffraction theory, and which is from 1925 or 1924, more or less. So in their theory, it's very, very clear for us that when you have a peak in your diffraction pattern, it's because you have a constructive interference between your scattering curves, your scattering waves, and if they have this kind of peak in that specific position, you may retrieve the distance between the atomic planes that give rise to that diffraction pattern, to that peak in that specific position. And it was amazing because we are in the 20s, the beginning of the 20s, the end of the 10th, and we are able now to measure the distance between atoms, using X-ray experiments with a very, very high precision, because we have a peak, very, very sharp peaks in very, very specific positions. And then when you have these peaks, for example, here I have this peak in this position, the second one is here. So the rate between this position and that one is very, very specific. And the rate between this one and this one, the third and the second is very, very specific. The fourth and the third is very, very specific. If you make the calculations, you must have to go to a table. This is a very, very simplified table, only for some cubic structures found in lipids. But we have hundreds and hundreds of possible structures, actually 230 possible structures that you have in tables, in crystallography tables. Then you must check, okay, I have this rate, and this rate between the second and the first peak, the third and the second, the fourth and the third, and so on and so far. And then you may, looking for this table, you can take a look, oh, I see that my ratios here are similar to those here. So I may infer that I have this crystallographic structure representing my system. This is what we call indexing peak process. This was very, very common in the beginning of the century. This was how crystallographers worked in that period, like 50, 60, 70 years ago, before computer time or these big tables and so on. So nowadays, it's kind of better to work with this problem, but we must keep in mind that what we need to know is the rate between the peaks, and then with the rate between the peaks, I am able to infer about the crystallographic symmetry or the crystallographic structure of my system. Okay? Good. So let's try to go to some more applied examples. I will talk first of all the simplest one, which is the working with liposomes or vesicles or lipid nanoparticles or something like this. And this is the first one that we are talking about today. When I'm talking about this kind of lipids in particular, I'm talking about lipids that prefer to go to perform this infinite plane. They have a cylindrical shape, and when they self-assemble, spontaneously self-assemble, they self-assemble in infinite planes. We, from the experimental point of view, from the laboratory point of view, we can change a little bit the sample preparation, and we can make, instead of these infinite planes, make like a round spherical particles or close to spherical particles, and with the same process. In that case, depending a little bit, this will change depending on a little bit of the diameter of your scattering particle. In this case that I will show to you, we are talking about diameters around 100 nanometers, which are 1,000 angstrom, 2,000 angstrom, something more or less with that order of magnitude. Now we must remember that the wavelength of X-ray is around the 1 or 2 angstroms, something from that magnitude. So I have 1 or 2 angstroms reaching at some nanoparticle which has 1,000, 2,000 angstroms. From the wave point of view, he's seeing an infinite plane. He's not seeing the curvature of the liposome because it's too high, too larger as compared to the wavelength of my photon. So in that case, we can make the math. Suppose that we have an infinite plane. It's the same math for us. So as a physicist, we like spheres and we like also planes. Because it's not easy, but it's less difficult to make the math to try to understand how is the scattering behaving in this case. So here, if I have, for example, this is what we should have in a liposome piece, a liposome surface. Of course, it's a schematic representation, but we can make it simpler looking for something like this. I have a parallelepiped. We have the X position, the Y position, and the Z position. And what is important is that the X direction and the Y direction, they are much larger than the wavelength of my X-ray. The Z direction, on the other hand, has more or less the same size of my wavelength, which means that from the X-ray point of view, I am only seeing the Z direction, Now, what we are trying to do, and there are several ways to do that in the literature, we are trying to make some calculations to see if we can get some information of my lipid organization. And please pay attention that the Z direction is the cross-section direction of my membrane, is the normal direction of my membrane. It's coming from outside of the cell or of the membrane to the inner part of the membrane. I am across the Z direction. And let's see if we can if we can make some calculations, see, and find the Z direction. We like to make models to this stuff, like the spherical column we talked yesterday. Now we have the plane, but I will try to improve a little bit this physical model by supposing that I know that the inner part of my belayer is hydrophobic. I have in the middle of the belayer itself a CH3 region. I have the tail is mainly CH2, methylene groups region, which has a slightly larger electron density as compared to the CH3. And the polar head group, which has a higher electron density. So I am split my bilayer into 3 parts, actually into 2 and 3 parts. First of all, 2 parts, I have a mirror over like this because I have a symmetry in this direction. What I'm seeing in the above of my leaflet on the bilayer, I'm seeing also in the end of to the other side of my leaflet. And here I have the CH3 region, CH2 and polar head, CH3, CH2 and polar head. To be better to you want to make easier to understand, is something as the lipid has this conformation here, this position. I have the polar head, which is represented by this blue region. I have the CH2 represented by this color here, and the CH3 region represented by that color, and everything is mirrored in the other side. Okay? This is the mathematical approach of this bilayer, and of course this is in all of that region. We are just making a geometrical model of our lipid. Why we are making this? If we remember the class yesterday, I mentioned to you when you have the geometry of your problem, and you know how is the electronic density distributed in your system, you can calculate everything analytically and compare with your experimental model. This is the geometrical model, and we use this model here to try to analyze the data that I will show to you in a while. So just to remember, we here in small angle X-ray experiments, we are trying to find the electron density profile or the contrast electron density profile, which is the difference between each space of that and the water. And we have this function here, is the electron density profile across the membrane. Only to make better you to understand that we have the lipids in this region. So here it should be, let's say, outside the vesicle. Then we are crossing the membrane. We are in the middle of the membrane, the CH3 region, CH2 region, polar head, and we are in the inner part of the membrane. This is a representation on what we have from that, on that point of view, on that symmetry. And that function here, you see, is the function that is describing my electron density profile across the membrane. This is the function that I'm looking for, because when I have this, I can characterize all my system and compare my experimental data with my theoretical simulations. Okay? So, we have these three regions. Each region will be characterized from the fitting procedure, from the fitting point of view, with two parameters. First of all, the length. I mean, how thick you have the CH3 region, the CH2, and the polar head, and the electron density profile of each specific region. The electron density profile, the electron density value of the CH3, CH2, and the polar head group as well. And we have the same parameters in the other part of the bilayer. This is the upper part in the inner part of the bilayer. So the thickness of the bilayer, which is also very important to characterize, is something like 2 times the sum of the length of each region of the upper part, for example, of the bilayer. And it should be something around 4 to 5 to 6 nanometer. This order of magnitude, if we are talking about regular and normal lipids. Perhaps you in your work, you work with polymer, with amphiphilic polymers, or amphipathic polymers. There are some polymers that may also have this kind of structure. They can form what we call polymersomes, which are this kind of somes, I mean this kind of structure, but composed of polymers. In that case, those values will change a lot because, of course, I'm using a different molecule with different chemical composition and different packing parameter, but will form very, very similar structure. You can use the same theory in that case, but the problem is that you must have some idea of those values in order to make the fitting procedure. Because you have to avoid to have too many fitting data, too many fitting parameters, and too few scattering curves. You may overfit your problem, which means that you may have several different solutions to the same experimental curve. And this is a problem. How can you be sure that solution 1 is correct and solution 2 is not correct if both of them have good fitting response? You must have, of course, parameters that have physical meaning. This is the first stuff. You must have reasonable parameters as compared to other data in the literature. So it's very, very important you to keep in mind that you must have some let's say, some indication of the order of magnitude of your parameters. When you do everything, you can make the calculation, and you will find out that here is the same stuff. You see, the scattering intensity here, I have the P of Q, but it's in our case, the same stuff, the scattering intensity, is the Fourier transform of this guy here. This is the same stuff that we saw yesterday 2 or 3 times. The only difference is now that we are in the Cartesian integration. And yesterday we were in the spherical symmetry, but it's the same stuff. You see here that I have a symmetrical integration from minus d over 2 to d over 2. D, of course, is the entire thickness. And, but when you are performing a Fourier transform, you must integrate in all the space. But for sure, from minus infinite up to d over 2, I am outside my vesicle, so I am underwater or whatever, and I have to subtract the electron density profile of water. So outside the membrane, this guy here will be every time 0. So I must have to perform the integration in the thickness of the bilayer. Then you can make some approximations, and you will It's not so difficult to arrive at that solution. It's slightly ugly, but it's not difficult at all. So you just have the sine function. And this is an analytical solution. So we can now try to fit our data using this kind of this particular equation. To do so, we use a software developed by Francesco Spinozzi from Ancona, named GenFit. And this is what we are doing to try to fit this data using the lamellar vesicle. So everything that I'm talking to you up to now is to show to you that we are tackle a specific problem. I started to work with these nanoparticles, and I have some collaborators, and we are working for the last 5 or so years in a slightly new, actually it's not so new, but I mean from the scientific point of view is new, research line that is named drug reproposing or drug repositioning area. What is the idea? The idea is that when you have a drug that was developed and tested for many years, 15, 20 years, to be treated for one specific disease, what we try to do is find a different disease that could be treated with the same drug. What is the interest in doing this kind of stuff? If I can use the drug that is already commercial, is already in the market, it's nice because I can avoid several years of clinical trials, of spending money in this kind of stuff. This is very nice. What is the problem from the commercial or from the pharmaceutical industry point of view? You may have some problems if you would like to make a patent for this kind of drug because as it's already commercial, you cannot make a patent for this drug. We are not caring about this, so what we are looking for is to get a drug, a well-known drug, use it for treat some specific disease, and try to use that drug to a different disease. In particular, me and my collaborators, we are interested in treat neglected diseases like malaria, like Chagas, like leishmaniasis, because those are very endemic here in Brazil, and we don't have so many treatments to them, and they are, when we have, they are not so cheap, or they are very harmful for the patient. So we are trying to find this kind of stuff. In this particular problem that I'm showing to you, we are using this Buporvacon, BPQ, this drug. This was a drug that was used in cattle to treat some kind of parasites. So we have this indication that could be used also to leishmaniasis. So we make a lot of effort. This my collaborator, he works with this Instituto Adolfo Lutz with parasites. We are also interested. We were interested in trying to locate the drug inside the bilayer. So this was the question that we would like to answer. Where is the drug in your bilayer? If it's polar or in the hydrophobic region. Then we make some small angle scattering experiment. This is in the abscess and presence of the drug. You can see that the scattering curves are very similar. And we have just a small difference here. And you may see that they have kind of a shift in that region over here. This was enough using that model that I have just introduced to you. This was enough to us to say that, okay, we may have some different kind of positioning here, which means that in the presence of the drug, or perhaps I could be able to locate the drug just using these two small differences. These are the fitting results in one case, and in the other case, the fitting parameters. We are not so interested in that. We are interested in this slide here. I already shown this figure to you yesterday. This, the dashed line here is the electron density profile of the membrane in the absence of the drug. When we put the drug, we see an increase of the function around here. We see an increase of the function around here. And we make several tests. And we were able to infer that from the small angle scattering perspective, from the small angle scattering resolution that we have, the drug must be located in that polar interface. Here, you can see that here I have the hydrophobic region. Here I may have water. For sure we have water here in the polar region. So this drug here likes to stay something like snorkeling the water and coming back, something like this. We have some collaborators that they are now trying to make some molecular dynamic simulations to try to show the same to try to observe the same behavior or not, or perhaps they can see a different position that we were not able to elucidate using small angle X-ray scattering. So I get this example to show to you because I think that it's important to know that if you'd like to get just a general information using small angle scattering like we made yesterday for some cases or some proteins, you can do that. You can perhaps sometimes just the radius of duration is the structural information that you are looking for, and that is enough for you to answer your problem. However, in some cases, you must have a more detailed description of your system to be sure to infer some specific property of your system. In our case here, to try to show where was the drug located, we must have several weeks and months of analysis of the scattering curves. Even I just show to you very, very quickly, it was not a very fast job. It's a very, very long job that we must perform. Okay? Some questions so far, so good. How are we? Fine?
Why don't you interpret the changes in the electronicity as a change in the lipid bilayer itself instead of distribution of the drug inside the lipid?
Actually, those two stuff, they are attached to each other. Because Sarkozy is an average technique. Since I have evidence that this is a small difference here, and the only difference that in one case I don't have the drug, in the other case I have the drug, I am inferring that I have the drug more or less around here. Of course, we have some other spectroscopic studies, evidence that our drug was inside the bilayer. We have this information using fluorescence technique, that the drug was inserted in the bilayer. So using that information and using this change here in the electron density profile, we can infer, we propose that the drug must rely in that region over here in the polar interface. But of course, it's possible to have other interpretations of the scattering data. There's no problem at all. This could be possible. We hope that those guys that are performing molecular dynamic simulations, they see similar behavior, but we hope. We don't know.
You want to do the experiments with living tissue?
We try to do that, but the problem with living tissue is that not necessarily. Every time you have double chains here, X-ray can damage easier as you don't have the double chain. When you get living tissues, several of the lipids, we have hundreds of different lipids composing the membrane of the cell. Several of them have these double bonds, those carbon double bonds, and they may have destroyed by your experiment. But there are several reports in the literature that people are using those lipids. They are trying what they do, they get the cell, they destroy the cell, they split the proteins and the lipids. They use those lipids to reconstruct, to rebuild, let's say, living-like system, but of course you don't have the protein, it's not living, but you have the same lipids that you have in the living system. And those lipids are not destroyed. You can make experiments on them. It's possible. It's possible. Okay? That depends on your lipid. In our case here, we are working with 7.4 because those lipids with the drug, those were used to treat infected mouses. Those mouses were infected with leishmaniasis. So we must have pH near to the intravenous pH of the mammals, and we try to control at 7.4. But the influence of the pH will be, of course, in the lipids and also in the drug. Perhaps your drug could be protonated or deprotonated, depending on the pH, and this for sure will change the interaction of the drug with the lipid. Those interactions have this hydrophobic contribution, but for sure they have also electrostatic contributions. Because the lipids that we use, they have deuterium, they have an electronic dipole in the polar head. So you don't have a charge-charge interaction, but you may have charge-dipole interaction, which is also electrostatic interaction.
There is any possibility to one layer penetrate into another?
Okay. There are some specific cases that you may have some kind of partial penetration between them, but those are not so often. And you can see then using X-ray experiments because you make the measurement and the total thickness of your bilayer is much, much smaller than those two lipids, for example. And there are some studies from a guy that used cationic lipid named DODAB in the presence with oligonucleotides. So you have a cationic surface with polyelectrolytes, very, very anionic. Then when you mix, you can sometimes you see very, very thin layers. They propose that you may have a partial interpenetration of the over, but this is not so common. Okay? So this was the first nanoparticle system that we characterize, and we are the liposomes. And the second one that we like very much also are the cubosomes. We started to work on that in the last 2, 3, 4 years, something around this. I already shown this figure to you. When you have, for example, this lipid here in excess of water, it tends to make structures like this. So we have these water channels. Here you have the one, and in the middle of those four, here, you have the second one, and among them, you have this kind of structure here, and you have this bilayer here. And they are very nice because they are quite interesting because they have a very, very hydrophobic region. For example, if you have a liposome, which is the inner part composed mostly from water, and you have a liposome and this double layer, you have more or less of 5% of hydrophobic region as compared to 95% of the hydrophilic region, which is the inner water pool that you have inside the liposome. In the case of cubosome, however, at the same size, you have more or less 50 and 50 volumes of hydrophobic and hydrophilic contributions. So what we believe is that the cubosome is a must. It could be a very, very nice nanoparticle to load with hydrophobic drugs due to excess of hydrophobic volume that we have in the particle. This is monolane, phytanthral. Those are their phase diagrams. Honestly, both of them, they are quite similar. They have a cubic structure in excess of water. In the two cases here, in the phytanthral, you may have also some hexagonal structure here when you hit a little bit, but they are very, very similar. And we use both of them, in particular, also to try to improve the stability. We use phytanthral because it does not have this double bond here that could be less stable as compared to the other one that has not any double layer. Sorry, double bond. I already mentioned to you that we have this lipid here. Those are known to make the cubic structure in the presence with amphiphilic also amphiphilic polymer. We can construct this kind of nanostructure to try to do the drug delivery. I will show a picture to you that it's better to understand what is the structure that we are believing we are performing here. And why are we studying these cubosomes? We are studying them because we'd like to put some drug inside. And the drug that we decided to work with was miltefosine. Miltefosine has a very, very particular shape. It's an amphiphilic molecule also. It's also able to form a micelles in solution. You can see you have a hydrophobic tail here. Those are methylene groups, no? So we have a hydrophobic region here. We have a zalterion here with this phosphate, this anionic phosphate, and this cationic choline. So I have this phosphocholine. And miltefosine is very, very known it's used against some kind of cancer. I think breast cancer, it's the most adequate cancer to be treated of, but it's very, very toxic. So the side effects of administration of miltefosine are very toxic to the person that is taking it. And on the other hand, I think in the end of the 90s, there are some studies showing that we have anti-leishmanial activity of miltefosine. So what we'd like to do is try to get miltefosine put inside the nanoparticle to decrease the side effects, but also to be effective against leishmaniasis. This is the main idea of this study that we performed in our group. And also in using the drug reproposing or repositioning methodology. I will not take time talking about the importance of treating leishmaniasis. It's a very endemic disease here in Brazil. I think we know a lot of that. And in all of those regions here in Brazil, in India, and in Mongolia in particular, we have some problem, but I will not take time talking about them. I will not talking about the production itself. I will talking about the structural characterization of them. Here we have a conventional electron microscopy. If you can say, for no, perhaps you cannot able to read, this bar here has 100 nanometers, so they have more or less 200, 250 nanometers. We have the inner part, just the liposomes. Here we have 1 micron, so they have half a micron, 400 nanometers, and so on. Here again, 100, and also here 100 nanometers. Then we have this particle with, let's say, some 200, 250, 300 nanometers that would like to make this drug delivery structure. This is their characterization. This is in the absence of multiphosine, just increasing the temperature. You see, those are the peaks that I was mentioning to you that characterize my cubic structure. When I have the ratio between the second with respect to the first, the third to the second, the fourth to the third, the fifth to the fourth, so on and so far, I can characterize all the cubic structure that I have here. And we can see that when we are increasing the temperature, we have other different structures. For example, here we have a hexagonal signature. Those are very, very typical from hexagonal signatures. And we have also some stuff over here, which we believe, according to the literature, that is a very, very strange phase that is also sometimes happens in cubic structures. And then we also have some, this is the lattice parameter. We'll see we are talking about hundreds of angstroms. So here 15.8, almost 19, almost 16, nanometers. So it's kind of large structures. And this is a quite nice advantage to this kind of nanoparticles, because we can try to use also a new technology that it's appearing, that there is already in the literature some examples, that is to use proteins to make the drug delivery. As some specific protein will be let's say your drug to be delivered in some specific region, depending of course on the function of the protein and so on. This is the cryo-EM images. When we have this cryo-EM stuff, it's very nice because, as you remember, the TEM images is not possible to see the inner water channels. Here you see the water channels. You can see several white dots. Each white dot that you are seeing there is a water tube that is going in that perpendicular direction as compared to the screen. We can also try to make some images, how can I say, some image modeling or something like this, and try to get some very specific peak positions, and we are able to get some repetition distances, and we can make also to get the lattice parameter of any particular cubic structure that we have in our image. What is different from SACs, from small angle scattering, is because in small angle scattering, I have an entire average of all cubosomes that are being illuminated by my X-ray beam. Here, we can see and choose each one of them I'd like to see how much they differ one to each other, or stuff like that. So this is a very, very nice technique that we have now, the last 5 or 10 years in Campinas. I'm not talking to you. Those are our results of cryo-EM. Those are images performed by Barbara. She started to work with this particular systems. This picture is quite nicely because in this particular case, when we see some specific particle, you may infer, please pay attention, you may infer some structure generally from your system. You see that here we have this water tube, we have this kind of globby stuff over here is what we believe is the polymer. So in that conformation, we believe that we have the inner cubic structure of our nanoparticle, and the outside region is composed by the amphipathic polymer that prevents these nanoparticles to self-assemble, to aggregate. Here we have small particles, we have larger particles, this one almost 400 nanometers, and this is nice also to see that we have a kind of a small polydispersion, and we may have different values of lattice parameters depending on your particular system. This is not a problem for us because, as I mentioned, small angle scattering, we have an average measurement, whereas here we have a very, very specific measurements from each specific scattering particle. Later on, we characterize also the general size of this scattering particles using dynamic light scattering. At this time, we were not so very good to produce the nanoparticles because the polydispersity was quite high, 0.3 now, but nowadays we improved a little bit the sample preparation and the cubosome production, and we get much, much smaller values of these cubosomes. Here are we are checking the influence of multiphosine. Here we have 1.5% as compared to the weight of phytantriol. And we see that we can destroy somehow the inner structure of the cubosome. We increase that part here. We start to have some kind of messy organization among lysozyme and phytantriol. Remember, multiphosine and phytantriol. Remember that multiphosine, our drug delivering system, is amphiphilic as well. So it may interacts with the phytantriol. And as I mentioned in the beginning of the presentation, this balance between hydrophobic and hydrophilic contributions, it's very, very delicate. So any disturb that you may have in this balance, you can destroy your cubic structure. This is probably what's going on here when we have multiphosine interacting with phytantriol. Nowadays, our collaborators, they are working on making the delivery of that drug, of that nanoparticle system in infected mouses, but this is not easy work to do because you must have several ethical approvals to perform this work and so on and so far. We are on that part right now. These are some examples of the latency parameters. And okay. Then we started to see, okay, but how much multiphosine we are able to load in our cubosomes. So we increased the multiphosine concentration up to 4%. At 4%, we have this curve here. And you see this is in the absence of multiphosine. This is increasing multiphosine concentration. And you see that even in the beginning of the experiment, when you have small amounts of multiphosine, you have this different structure here, which was very, very similar to the one that we observed when we increased the temperature of the other cubosome. And those are the surfaces. And this is the structure of these particular peaks here. It's a very, very peculiar and strange structure because it's composed by micelles against with some kind of tubes, hollow tubes. So it's a very, very complicated system to be analyzed. We are also discussing this result a little bit with my former supervisor when I was in Italy. He worked a lot with that system. He made a postdoc regarding this kind of system. We are trying to discuss a little bit which could be the reason that we have this kind of structure, even at small concentrations of drug. Then we use a dynamic light scattering and nano-tracking analyzer, NTA, to try to get the information of our particle stability and concentration. So this is what we have we were trying to improve the procedure of producing the cubosomes, so we tried different stuff like lyophilization, like structurization, and we just to try to improve our way to perform the cubosomes, to perform our nanoparticles, to be sure that we could deliver to our collaborators very nice and stable particles, and those are stable for at least 1 month. This is what we evidenced. But there is not only us making this kind of study around. These studies from Esposito and collaborators, they are trying to load inside the cubosomes another drug named Curcumin. Curcumin is a very well-known dye, used even to cook. And they have this kind of cubosome, very, very nice pictures that they observed. And you may see that you can have, along with the cubosome, different kind of structures when you try to mix this kind of things. This is common. We don't have a kind of, let's say, clean system. But the main important thing is that this system here is loading curcumin according to the authors, and they see a shift change from the cubic structure to hexagonal structure. These 3 peaks here, they are very, very characteristic of hexagonal structures. Why I'm saying this? Because if you do the rate between this guy and and this guy and this one, you looking for, if you looking for in the crystallographic table, you find that the hexagonal structure is the one that fits this ratio. And also, they used chloroform too, because it's a very, very insoluble dye. They use chloroform to try to load this in the cubosomes. We have also some data indicating that depending on a little bit on the organic solvent that you are using to characterize your system, you for sure may change the inner structure. For example, in decreasing peaks. When you decrease in peaks in a scattering curve, you are decreasing the crystallinity of your system. You are decreasing or the size of your entire system, or the range of your crystallinity. And even when you lose the peaks, you don't have more. You don't have any longer your cubic structure. Perhaps in that case here, you have an isotropic micellar system. Okay? So these are, I would like to talk these two topics to you. And I decided to talk a little bit about two techniques, but I think I will talk just about cryo-EM. I will not talk about the dynamic light scattering because it's a quite common technique. And well, we don't have, I think, enough time. So we got in touch with the cryo-EM technique around 4 years ago, when they installed the first microscopes here in Brazil, when they bring the technology here to Brazil. It's a very, very nice and powerful technique. Use it to characterize not only nanoparticle, but any kind of nano-sized system. I will skip this kind of stuff. When we are talking about electron microscopy, we have two types of electron microscopy: the transmission electron microscopy and the scanning electron microscopy. The transmission one is when, as the name say, your electron must cross your sample and you detect them after crossing the sample. And the scanning one, you have the reflection of the electrons, and you get the electrons that do not cross your sample. Here's a pictorial representation of an electroscopic system. The main idea here is that you have in physics the principle of the duality of the light, for example, if it's a photon or if it's a particle, and also from the electron point of view, that sometimes they may behave as a particle, and that sometimes they behave as a wave. And they behave as a wave when they have very, very high speeds, very, very high velocities. So here we accelerate them a lot, so this beam behaves just like photons. And what is more interesting for us, according to the de Broglie principle, their wavelengths are very, very smaller as compared to X-ray wavelengths, for example. So we can use them to see nanoparticles that have sizes much smaller than the visible light, because the light that we are using to see, which are the electron waves, are much smaller in the wavelength. This is the main idea. This is why we call electron microscopy, because our waves are now the electrons. In the cryo-EM, the most important thing from the cryo-EM as compared to the conventional electron microscopy is the sample preparation. They are able to freeze your sample in such a way that the conformation is the same as it was in room temperature. This is very, very weird the first time we listen. Don't worry, we will get into this in a couple of slides. This was so huge, so important from the structural biology point of view, from the nanomaterial science point of view, that the three guys that developed this kind of stuff, there are several other researchers developed this. They choose these three guys. They won the Nobel Prize in 2017, I mean, 2 years ago. I say 2 years because the Nobel is laureate in the end of the year, so the last one was 19. And they started, Joachim Frank started to work with analysis, from sample analysis, to get the micrographies and try to make some kind of shape reconstruction. And Dubochet studied, he was able to vitrify water. We will talk about this in a while, but this is very, very important for the cryo-EM. And Henderson was the first one to use electron microscopy to get some information of virus. And Henderson will be with us here this year in our meeting at UPAB that I mentioned to you, which takes place in October at Foz do Iguaçu. He will be, I think, the last lecture To close the entire meeting will be from Professor Richard Henderson. The vitrification process is the most interesting and fun and nice part of the cryo-EM. The main idea is you will freeze your sample, you will freeze water in such a way, and it will be so fast, but so fast that the water does have time to rearrange itself, so it's keeping that position. The first time that the person explained this to me, they used these exactly words. I say, I don't believe you. I say, why? Because water is a very, very small molecule. It changes a lot. How fast you must freeze some stuff that the water itself, it's a very, very small molecule, does not have time to rearrange. And I said, oh yeah, but more or less. And then I start to study a little bit, and I see that the guy was not wrong. I mean, the explanation is more or less like this, but there is a lot of very beautiful physics behind of that. This is if you are a physicist, you have some nightmares with this guy here. This is the water phase diagram. They say, in physics, we call that water is a simple mixture, a simple substance. Water that has nothing of water that is simple. It's a very, very complicated model. There's a lot of interaction and behavior and a lot of stuff. This is what we call the triple point of the water, that we have liquid with water, with vapor, with solid water, and so on and so forth. What is important is that when you vitrify waters, the first stuff, when you get ice in your home, in your place, the ice has an hexagonal structure. It's hexagonal structure. You see also some snowflakes, the hexagonal position. It's more or less like that. When you decrease even more the water, they have a cubic structure. This is around -8. You have from the hexagonal to the cubic change. I'm talking about the room ATP conditions, ATM conditions, sorry. However, there are some specific liquids that they have a thermodynamic property, which is the heat capacity, the CP, which is from the physical point of view, is the capacity of that element to exchange heat with someone else. For example, if you wake up in the morning, you have your room, all the temperatures more or less equilibrated and so on, but when you put your feet in the ground, you feel cold. Of course, the ground is, let's say, 22 degrees, 23 degrees, and you have 37. Okay, the skin is a little less, but don't worry. When you put your hand in, let's say, in the wall, you don't see that cold, but the temperature of the wall and the temperature of the floor are the same. Why? Because the capacity of the floor to exchange heat with you, it's larger than this one. It's not that one is warmer and the other is cold or something like this. It's the ability of that element to exchange the heat with some other. We have two liquid substances, liquid ethane and liquid propane, that have very, 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 very high capacities. They have, they can exchange very, very, very fast their heat with some other substances. Much more than, for example, liquid nitrogen. Much more, much more. So what that guy, the Bush realized is that he could, for example, if he had a very, very, very small volume of water and a very, very large amount of liquid ethane, because of the mass of the water will be very, very small, the capability of exchange heat will be so high that water could be vitrified, which means I freeze the water without making ice, without giving the water time to rearrange. So he developed this vitrification process, and this was the reason that he got the Nobel Prize against for the other two professors. And we use liquid ethane to do this process here nowadays. And to get an idea, the liquid ethane has a temperature around -80, -85. If you remember, liquid nitrogen is almost -190 degrees. Almost 100 degrees smaller temperature as compared to the liquid ethane. Nevertheless, the ability of the liquid ethane to exchange its heat with water to steal the heat of water is much higher than the liquid nitrogen. So if you try to do vitrification in liquid nitrogen, it will not work. you don't have vitreous water. You must have a liquid ethane to make the vitrification. And this is funny because when you have the vitrification, water, the vitrified water hits a temperature, then you can move that to liquid nitrogen. There is no problem at all because the water was already vitrified in liquid ethane. This is the procedure that we do. Then now you have this specimen, then you go to your microscope, and you must, of course, ensure that the temperature of that grid will remain as smaller as it has to avoid the water to come back to get movement again. This is the vitrification process, and this was which helped a lot the researchers to work with the cryo-EM methodology. So after you vitrify, you have your samples, you vitrify your sample in grids, what we call grids, something like this. To have an idea, this guy here has 3 millimeters of diameter. You have, so here, your specimen, your sample, and it's vitrified. Then you go to the microscope, you start to align the beam and so on and so forth. And then you have this kind of stuff over here. In this bar here, you have 100 microns. So those are very, very huge. Of course, you must get a close in one of these. You have stuff like this. Now we have more or less 10 microns. This is a very, very common, what we call carbon grid. Here you see that we have a hole. It's very, very common to have several fails inside our grid. But what we need to make our analysis, just a couple of good pieces in our grid. It's fine to make some conclusions. We can see that we have some fails here, some problems, and also here. Of course, to remember, this is a transmission microscopy. So when I have this white stuff, it's because the electron is able to cross and is detecting the detector down the grid. Here, of course, I'm not seeing anything because those grids are composed by carbon that absorb a lot. The electron is not able to cross those carbons. And then we must analyze only those holes over here. And that's not important for us. And then, okay, now that we have to observe our holes, let's say we get very, very good images of your system, we now try to make the reconstruction of the sample of our system. The first stuff we try to do is the cryotomography. We are starting this in Brazil right now. So we are able to get the grid to title a little bit in both directions to get information of both directions. And then with that information, we can get stuff like that. This in the left part was the first time that we get some time of the biological, structural biology point of view. No? The first time they have some kind of shape reconstruction. And here is the same structure made it several years after that when we increased a lot the softwares that make this analysis, the quality of the detectors, and so on. Then we can improve a lot the resolution or what we have. Nowadays, we can go even better. We can get even more nice details, even more nice reconstructions of our system. Of course, to do that kind of stuff, it's not possible to use any regular microscope, as we have dozens in Brazil, but we have to use very, very specific microscopes. The most famous are those from the cryo family, from this company, the Thermos company. In Brazil, we have a couple of them here we have, one Titan and one another is named Arctica. Here at Campinas, they are there to, then we can use and try to get this structural information. They have several possibilities. For instance, it's possible to use those equipments. Then you can load, you see, several grids over here. You can load up to 11 grids. This means that you can do 11 different studies at the same time. Those are very, very automatic systems. So you can program the electron, the microscope to work for, let's say, 1, 2, or 3 weeks, straightforward. It's working, they change itself. Of course, you must check once in a while because perhaps you have some problems during this procedure. But it's possible to work with this cassette here with 11 grids to be measured at the same time. This is an image from here in our lab. This is inside the microscope. Here we are turning on this, for example, is the Arctica 1. So okay, then we have this possibility. We can now make the grid. We know how to load the grid, how to get everything, and how can we get from a tomography, from an X-ray, a reconstruction of our sample. Actually, this is what we get when you go to the doctor and you have something, let's say in the lung or in your leg. He asks you to make X-ray tomography. You do something like this. But when you go to the cryo-EM to make some experiment, you have something like this, the same image. You see, it's almost impossible to see a difference from the system to the other. This is because if we, in order to change, in order to see this, to better see this stuff, you must get out from the focus of your lens, of your beam, and when you go out of the focus, you lose the resolution. As near you was the focus, higher is your resolution. However, if you are exactly over the focus, you cannot see anything because you don't have any information of your electron waves. So those images, this is why we have several different kinds of softwares and algorithms that are being used to try to reconstruct the samples using this kind of information. The idea is that when you do several thousands of these images, this is your sample, but you may have different projections. You have different Fourier transformation from the different projections. Then you must somehow try to combine them in the Fourier space. Then you come back to be in your Fourier, to get your Fourier information again. Of course, when we make the measurements, we are making those measurements here. Okay? I don't know this a priori. I get this information. I have to what we call classified my system, which is to split every image that has that conformation, every image that has that conformation, and every image that has that conformation. Then I go to the Fourier space. Then I go to the shape reconstruction. This is another example. This is what our image looks like. So you get this. You try to split in different classes, and then you try to rebuild, and then you get the information. This is why we get a very, very high resolution. For example, I can infer to you that this domain here is in that part and is not here. So I can fit that guy here inside this electron map that we made. So the size of this electron map is more or less compared to our resolution. When we talk that we have 3 angstrom resolution, I can say that the position of that guy is here with an error of more or less 3 angstroms. It's more or less like this is how we talk of resolution. So this is an image, so we have to work with stuff like that to say, well, Professor, I don't see anything. Yeah, it's difficult to believe. This is why we must have these computers working for us. And this is also the reason that we must pay a lot of attention, because sometimes computer makes mistakes, and then you can make some analysis, and this is not what you are wondering that you could see. This is a nice image because you have different kind of structures, but when you try to classify them, you see this one is similar to this one despite a rotation, which is similar to this one despite another rotation, which is similar to that one despite another rotation. So what the software does, he gets this kind of information, he rotates those images so they are one above each other, and then he makes some kind of average. This is called the classification process, and then you have some stuff like this. So this was a classification after thousands and thousands of images. So we can reach this 16 or something like this different classes, and now you can try to get this shape reconstruction of your particle. It's kind of a tomography problem, and you get the 3D image again, your 3D image. In that case, it's something similar to that. So you see, now when we say that the cryo-EM is able to give you very, very high resolution images, we are saying that I can be sure that this residue, this lateral chain of this tryptophan is in that position within a 2.5, 3, 4 angstroms precision. This is the high resolution that we are talking about when we are talking about cryo-EM for single particles. We are not talking any longer about the nanoparticle cryo-EM measurements, but single particles cryo-EM or shape reconstruction using proteins. Of course, I talked to you in 15 minutes, 20 minutes, but this is a very, very hard job to do. They are in Brazil, just a couple of researchers, of course, a couple, I mean, 6, 7, 10 researchers working on single molecule shape reconstruction, because this improves a lot the possibility that you may have to get the crystallographic information of your protein on your work, but it's not a job. I mean, we are talking about 1, perhaps 2 years working here from the sample, if the sample is good, if the sample is monodispersed, it's good, and so on, because you stay a lot of time trying to improve the sample preparation, because if you have a lot of fails in the sample preparation, you will not be able to get the thousands of images that you must get, that you must have. So it's a very, very tough and not easy job. And then you have to do the assignment of the regions from the residues of your protein and get the information. I wanted to show to you a final example of using cryo-EM to get this kind of stuff. And this study here is not, this was from 2 years ago, 3 years ago. They are studying the influence, they are studying the polymorphism, I mean, the different forms or morphologies of Alzheimer, of amyloid fibrils for Alzheimer brains. Those are their images, you see? And this is what they are proposing. They are proposing to have 2, perhaps 3 different arrangements. You may remember yesterday when I talked to you about neurodegenerative diseases, I said, oh, all the proteins have the same mechanism of action. You have the protein, but somehow they have a kind of polymerization in a very specific structure, cross-beta structure, and everything is the same. The cross-beta from Alzheimer and from the Parkinson, from the crazy cow disease, they are all the same. This is true, more or less. What they are shown here 2 years ago, that they may have some differences, but I mean, it's a very, very detailed study, so it's a very, very nice one. They use the cryo-EM to get this kind of information. And here, after doing all of those reconstructions, they were able to see exactly where were each of the residues of these peptides, these proteins, and also to elucidate who is interacting with whom here inside to see how can you have this cross-beta structure. Here I have one peptide, down here the second one, and here is the residue that he believes is the most important for that interaction. Even in that study, he also saw the influence of lipids, some different lipids in the cryo-EM of this. Now, sorry, this is another study in alpha-synuclein of Parkinson, because there are some evidences that you may have also along with the peptides in the peptides in the membrane, along with the peptides in the amyloid fiber, you may have also some lipids composing these amyloid-like fiber structures. Okay? Guys, questions? And ladies? Yeah.
About the reconstruction.
Mm-hmm.
Like, can you give a little bit
Sure.
how the program does? Because I think before. Yes, because if you talk about the rotation and stuff like this, but what parameters is used? Because if you try to compare everything with everything,
No, this is very difficult. This is why those algorithms, when they were first developed, they were very, very, very important, which also the first guy that won the Nobel won by this. If you pay attention, there were 3 guys. The first one who make the algorithm was the first one. He get half of the prize. So in their opinion, this is the most important part. Then the other half, we split into the vitrification, which is also very important, but not as compared to the first one, and the first guy who solved the first virus structure in 1990. So I have 3 guys, but it's half for one, and 25 and 25 for the other guys. Because this is very difficult. It's not easy to work with image, in particular with image that it's not easy to split the contribution of the image from your background. You must have you must make several images. You must recognize and make several images. When I make several, I make hundreds and hundreds of images from your background. Then you make a very, very good average of your background. This is your first and more important, let's say, starting point. Because when you have a good average of your background, it's easy It's easy. It's less difficult to try to differentiate what is background from what is information. And then, of course, as we are improving the software in the computer hardware, we can increase our capability to work with more larger images or more detailed images. We are also increasing the detector. So nowadays, we are working with what we call 4M detector. Then we have a pixel, millions of pixels that get in the information. And we are improving this kind of stuff. But the algorithm to do that stuff is from the 75, 76. But it's stuff like this. You try to recognize who is similar to whom. Then you try to align them. But you have to recognize those who are similar. Then you align them. For those who are not similar, you don't align people that are structured that are very, very different. And this is the most important thing. This is why this is a very, very long and hard job to perform. It's not easy to do that.
The electron beam is a thermal beam, right? So there is no coherence. And therefore, there is a phase problem.
Yeah.
And to solve a phase problem, you need extra information from outside. So is it true, I would guess, that they already know that the ATP synthase has two domains that are connected. Mm-hmm. So they have to try to use this information to improve. Is that correct?
Yeah, absolutely correct. Actually, here is the first step of a very, very long chain of information. I mean, this is perhaps after 5 or 6 years of working when you get a polypeptide sequence, for example. You must you must have a lot of information, for example, chemical information. Which residue is near to which residue. This helps you a lot here. If you don't get that information, just like X-ray crystallography, you will not be able to fit the structure with that resolution. You must have that kind of information. This for sure. This is the end of the step. I mean, this is the end of the chain, let's say. If you don't have that previous information, perhaps if you don't have those informations, you may not even want time to make the measurement because cryo-EM is a very expensive technique. To have an idea, 24 hours of this Titan, which is the most powerful microscope that we have nowadays. And to do this measurement, to get this kind of information, perhaps you need 4 or 5, 1 week. Each day costs 3,000 euros. 3,000 euros per day. So to do that information, to do that measurement, you must have several information a priori. Otherwise, they will not even allow you to make the measurement if you don't have this kind of information. This is like the refinement of perhaps a previous structure that have a 15 angstrom resolution or 20 angstrom resolution, very, very loose, but you must have those information.
What is the cost of each of those microscopes?
Oh, the last one is the most expensive one, around $6,000, $6 million, $6 million, $6 million. Each. You have some, I was, for example, at Stanford, they have 4. 6 each, yeah. And it's funny because, okay, it's different way of work, but you have some electron microscopy centers in the world, those are amazing. And as far as I know, there are only 2 that are free of charge. Here in Campinas and in Diamond in England. Those are free of charge. But to use in Diamond, you must reach there having something like this. You have your system already classified. Then you can use the microscope to refine and try to build this kind of structure here. If you don't have this kind of stuff, perhaps they will not give you time in the machine. But every time you must pay something to use. But here in Campinas, in Diamond, as far as I know, you don't need to pay. You'd like to ask me something?
Richard, wondering if you apply some in software, applying statistical analysis like SDA.
Oh, I'm not so sure about this detail, but I mean, of course, you have some very, very serious statistics behind this kind of analysis. But which kind of analysis is the PCA or I'm not so sure. I don't know that. We must ask from Rodrigo Portugal that he is used to work with the single particle systems.
It's not
Yeah. Who is working a lot with that kind of clusterization problem is the Professor Marijn van Hel. He was the former director of a cryo-EM center I think, in Imperial College in London, if I'm not sure. And he was invited to come to Brazil, and he's in Brazil helping us to try to build this cryo-EM community here. And he's helping a lot the guys from the center, because nowadays here in Brazil we have a Titan, Antarctica. We have more or less, it's borrowed. Another one is the Tylus one. This is the first, the second better, the third better one. So we have all of them. And we have a conventional one, which is also able to make cryo images, but with not so good resolution. So this is good for nanoparticles, like the Cubosomes that I showed to you, where the angstrom precision is not important for me. But for protein, you have both Tylus, Arctica, and the cryo. They could be used to get some kind of information. Of course, depending on your problem, you should go to one microscope or to the other, but these are the technicians from the lab. They know how to guide you in that regard. Some other question? I didn't understand, sorry. No. Actually, I didn't talk about this slide for you, but this is, what is the problem? Let's say that this is the object that I'm studying. Let's suppose this is the protein, these 3 dots here. Depending on the way that you look, these 3 dots, you may have something like this, if you imagine an infinite cylinder. Something like this. And depending if you have different positions, you can have something like this. For example, these 3 here, those have this alignment. If you rotate, similar to these 3 here. If you rotate again, similar to those 3 here. But they are rotated one to each other. Then you do this kind of process to try to reconstruct your sample. This is also just an example on how this the Fourier transform and helping you to try to do this kind of shape reconstruction analysis. Just a general example, because it's not so easy to understand this kind of going to the Fourier space and coming back. It's just an image treatment. Guys, if I can, just would like to remember you for the 10th time, because it's important that, because we are having several this one. Just to remember you that we are having these 2 meetings here organized by the Biophysical Society, and I think we will have very nice presentations there. We have several. From this one, unfortunately, not so much, but from this one, we have several different fellowships to bring you, students, young researchers, to the meeting. If you are Brazilian, if you came from abroad, you have several options. Please go there to the website, because some of them, the deadline is not so far. And if you have some doubts, you can write me as long as you want. And thank you very much for your attention. Você vai dar algum recado, alguma coisa? Não? Pessoal, então a gente volta então às 2 horas, não é isso?